Good day to both Suba and everyone. Our group member consists of Rui Yue, Ooi Tze Yong, me, Shian, and also Kelly. So the topic for our presentation is forensic DNA phenotyping, dog breed classification, and skeletal phenotype prediction. So I'll start off with the introduction. Forensic genetics is the branch of genetics that related the application of genetic knowledge to legal problems and legal proceeding. It's also a branch of forensic medicine related to application of medical knowledge to legal matters. So in animal forensic genetics, it's broadly divided into two: domestic species and wildlife. Domestic species usually cohabit with the same environment as human. They can be used to identify the crime perpetrators or to clarify cases of animal attacks on humans or other animals. Whereas for wildlife species, they are normally will be used to identify taxonomic source of non-human biological material, either as agent to trade or direct or indirect evidence in other crimes. So classification of unknown canine individual according to its breed is performed using molecular markers that exist within the nuclear DNA of the dog genome. The genetic marker for purebred will match with known reference dog breed genotypes and shows strong concordance with the predicted genotypes at the loci of interest. And for the mixed breed species, genotypes of mixed breed dogs at loci of interest match with multiple reference population. The degree of match depends on various factors such as ancestry of its parents and genetic recombination. So there are two genetic markers which are currently used, which are short ternary repeats, STRS, and also single nucleotide polymorphism, SNPs. So STR, short ternary repeats analysis, is a widely used technique in forensic genetics for DNA profiling. The process of STR will start off with sample collection from unknown species, then amplification of the DNA sample using PCR to create multiple copies of the STR region of interest. Electrophoresis is carried out to separate the different STR regions, and the last, the comparison of the STR factors of the unknown sample to a reference database of known species. SNPs array are a type of DNA microarray that are used to genotype individuals at hundreds or of thousands to millions of SNPs in the single array. The labeled DNA sample is hybridized to the SNP array, which consists of a grid of DNA probes that are specific to certain SNPs. Those allelic-specific probes, which matches with the target DNA, will form hybridization, whereas those doesn't match will not form hybridization. So after hybridization, the array is scanned to detect the fluorescent label on the bound DNA, and data analysis is carried out to determine the genotype of the individual at each SNP locus. So next, I'll pass to Rui Yue.
So I will continue on the forensic DNA phenotyping FDP. So the FDP could replace the use of STR and large commercially available SNP arrays for better identify specific and external morphological features of a canine, which is the dog breed, when the identity of the canine is unknown. FDP can also predict externally visible characteristics from forensic DNA trace. So since the difference in morphology of the skeletal system are highly visible for untrained witnesses, so they are very suitable for assessment using this FDP. So differences in skeletal morphological of the dog breed allow them to be easily differentiated visually, for example, through their body size, body shape, skull shape, limb length, and tail morphology. So in this case, ears are also easily identifiable external trait. So the use of genetic marker that characterize this skeletal difference among domestic dog breed will be very practical and easily translatable for the forensic investigator. Therefore, SNP and indel marker selection should be targeted to these loci that drive these clear phenotypic differences. So these are the genes variants for the characteristic of body size, where the small and medium sized dog, these are some example genes that associated with the body size difference, including the growth hormone receptor GHR with two genetic variants, GHR1 and GFR2, and also the IGF1. Then these are the genes with genetic variant that are close to fixation or almost unique to the large dog population, including the ZNF608 and IGSF1. So studies show that the genes, for example, ACSL4, have been associated with the body shape differences among the domestic dog breed. Then in the skull shape, two genetic variants that may drive skull shape difference are found, which are the BMP3 and SMOC2. Then a factor that determine the ear shape across the domestic breed is the shape of the auricular cartilage of the dog. So the gene variant associated with the ear shape are found, like for example, in the exogenic region of long intergenic non-coding RNA with the WNT inhibitor factor 1 and the methionine sulfoxide reductase B3. So scientists also found that the FGF4 retrogene can cause the growth plate in the long bone of close prematurely. So this will result in the observed disproportionate dwarfism phenotype. So in the digit type, which is the hind limb type, variant in the gene, for example, ALX4 and SHH gene are proposed to be responsible in this formation of the dog breed limb. Then lastly, the tail morphology gene, for example, DVL2, is used by the forensic investigator to differentiate dog breed with a screw tail from other domestic dog breed. Then I will pass it to Kelly for our presentation.
So now let's focus on how they apply the skeletal phenotype genetic markers for group assignment and phenotype prediction. In this paper, they group the dog breeds using large, small, bulky size of marker, as well as the phenotype-based markers to derive the allele frequency of genetic variants. They tested the markers to separate 10 most popular American Kennel Group AKC registered dog breeds into groups. This table This table shows the derived allele frequency of genetic variants at large size markers, and the markers that highlighted in green are those that can differentiate the dog breeds. As we can see, Golden Retriever, French Bulldog, English Bulldog, and Rottweiler can be differentiated as a group through the genetic variants at IGSF1, while German Shepherd, Standard Poodle, Rottweiler, and Pointer can be differentiated in a group through LCORL. If we look at the small size marker, only miniature and toy poodle can be differentiated through the analysis of variants within HMGA2 and IGF1R, while the derived variant within other markers are not useful because the differences within the values are small and we cannot differentiate them clearly. Same goes to the bulky size marker. Only Rottweiler can be differentiated from other AKC breeds using ACSL4, and the variants within H and F4G are not useful. If we look at the phenotype-based marker, they tested on the markers of skull shape, leg size, tail shape, and tail morphology to group the dog breeds. So they grouped them based on the significant difference in each marker and allowed them for phenotype prediction. Since we got the markers already, we now can look at the assay development to detect non-skeletal genetic markers, which is competitive allele-specific PCR, KSP. KSP is a scalable genotyping method that can be used to detect and differentiate SNPs and indels at the specific loci. It requires two allele-specific forward primers that can competitively bind at the loci of interest and a common reverse primer. Then 3 groups of PCR will be done, using 1 primer for each and also both primers simultaneously. The tail-forward primer contains a fluorescence resonance energy transfer cassette, allowing for endpoint fluorescence detection. So we are able to differentiate the sequence with both primers' fluorescence to determine the genotypic profile of a dog at the loci of interest, and then allow the forensic investigator to predict the numerous skeletal phenotypes of a dog. Then we can, that's all for me. We now move on to the further challenges by Tze Yong. Thank you.
Thank you, Kelly. So I'll proceed the presentation with future challenges. For the first challenge, is the mistakes in breed attribution might occur at any gene variant that is not close to fixation. For the second challenge is that a lack of clarity in the phenotypic descriptions of the morphological traits such as the ear shape and the length of hindquarters may create the confusion in breed discrimination. For the third future challenge is that the connecting genotypes with the phenotypes from the same individual through the application of high resolution phenotyping is very costly. This will limit the experimental sample size available. Next. The fourth challenge is that the further validation of the genetic markers with larger sample sizes in relevant dog populations is needed to determine whether the levels of fixation proposed in prior research reflect actual levels of fixation in modern breeds and different geographic subpopulations of the breeds. For the last challenge is that developing in an accurate model to predict the size of a dog is very challenging. The phenotypic effect of each derived size genetic variant needs to be assessed according to whether one or two copies are carried. And currently, the modeling of genotypic combinations of the derived and ancestral variants is used, but this approach is not very practical because 59,049 possible unique genotypic combinations exist between the 10 allelic size markers. And most of these unique genotypic combinations will probably only realize in some mixed breed dogs. Next, for the conclusion part, the current genetic marker approach to classify domestic dogs is forensic DNA phenotyping, which is FDP. And this is because the domestic dogs display a spectacular array of morphological differences that are driven by a few loci with SNPs and indels of large effect. Using DNA evidence, forensic investigators can determine the breed by quantifying the phenotypic effects within the breed to discriminate the individual dogs depending on the skeletal phenotypes of interest. And furthermore, to have a better breed classification, alert frequency estimations of the skeletal markers need to be undertaken in a larger cohort of dogs so that their fixation levels within the breeds can be more accurately gauged. And for the next slide is our reference. So that's all from us. Thank you.